Formation. What is urine? Urine is excreted from body as a waste product, right? The renal system is responsible for the excretion of waste product from the body in the form of urine, right? Our renal system, urinary system is responsible for the formation of urine. First of all, urinary system excretes the waste from body in the form of urine, right? This urine is composed of, what is the composition of urine? Urine will be composed of water. Up to 95% urine is composed of water. This urine also has some of electrolytes: sodium, potassium, magnesium, calcium, right? And this protein also have some drugs we are taking for the treatments, drug toxins. Also this urine contain some toxins which are made from metabolism of the nutrients which we are eating. Also you have urea in urine, and urine is called urea because this urine is called urine because of this urea, right? So this is the composition of, generally this is the composition of urine, and also it has some other waste products from body, right? Today we are discussing how this urine is formed in kidneys or in nephron. The processes which we have discussed previously: the absorption, reabsorption, the secretion in the proximal convoluted tubule, the filtration, the glomerulus, and distal convoluted tubule, the reabsorption of water. All these processes are taking place for urine formation, to excrete the waste product from blood into urine, and in urine it will be leave the body, right? So now we are starting the urine formation. Urine formation occurs in 3 steps. Urine formation completes in 3 steps. First step is glomerular filtration. We already have discussed this in detail. Glomerular filtration. It is also known as ultra filtration. The second step is renal reabsorption or tubular reabsorption. And the third step is tubular secretions. It is also known as active secretions. What is the difference between reabsorption and secretion? When the materials are transferring from the nephron to blood, this is called reabsorption, and when they are transferring from blood to nephron, this is called secretion. So for example, I have, generally this is for example, this is nephron and this is collecting duct. From here we have glomerulus. This is glomerulus. The first step occurs here when the things are filtering from the glomerulus, the fenestrated capillaries, diaphragm, the podocytes, right? These are the barriers. Are also the glomerular basement membrane is responsible for this filtration. So the first step occurs here. This is called glomerular filtration, right? The second step, for example, these are peritubular capillaries. These are peritubular capillaries, right? Here in proximal, this is proximal convoluted tubule, this is loop of Henle, this is distal convoluted tubules, and these are collecting ducts. The things will be transferring from this proximal convoluted tubule into the blood. This is called reabsorption. This reabsorption occur here also, but in loop of Henle, descending limb, only the water is reabsorbed, and this is the ascending loop of Henle, and here only the chloride and sodium ions are reabsorbing. So this step is called reabsorption. Reabsorption of materials from the lumen of nephron into blood. And some of the things, for example, drugs, toxins, urea, these things are secreting from blood into nephron. So this is called secretion. We have discussed and detailed these processes. Today we will only take a look on these processes and will repeat, and will in last we will see that how urine is formed under the processes which we have discussed in proximal convoluted tubules, in glomerulus, in loop of Henle, distal convoluted tubules, and collecting ducts, right? So for example, I have this nephron. This is the glomerulus, and glomerulus is composed of podocytes. This is Bowman's capsule, and Bowman's capsule is composed of podocytes, and they also have the visceral and parietal layer of epithelium, right? This is, for example, Bowman's capsule, and glomerulus is placed here. For example, generally this is the afferent arteriole, and this is the glomerulus, and this is the afferent arteriole. Right? And I have discussed that this epithelium is composed of fenestrated epithelium. This wall is composed of fenestrated epithelium. What is fenestrated epithelium? That this have a lot of pores. Do you remember this? A lot of pores, and therefore it is called fenestrated capillaries. Right, and here we have a very special basement membrane is placed, and this basement membrane is called glomerular basement membrane. Right, the step occur here in urine formation is the ultrafiltration, or it is called glomerular filtration. But this filtration occur under some pressures. Under some pressure, we have net filtration pressure. Do you remember that? Glomerular hydrostatic pressure, colloidal osmotic pressure, and capsular hydrostatic pressure. Let's remind that pressures. We have net filtration pressure. This net filtration pressure is equal to pressures which force the glomerular filtration, right, and minus the pressures which prevent the glomerular filtration, right. What are these pressures? Pressures which forcing. So here we have glomerular hydrostatic pressure. The blood itself is forcing to filter from here, and this is called glomerular hydrostatic pressure. It forcing the filtration in glomerulus. What is glomerular hydrostatic pressure? 55. This is 55 mm of Hg. So glomerular filtration pressure is 55 mm of Hg. Right? The second thing, we have another pressure here, but proteins are not filtering from here, right? And here the protein, we well mentioned that proteins also forcing this filtration, and this is called glomerular capsular osmotic pressure, or this is called the colloidal osmotic pressure. Colloidal osmotic pressure in Bowman's capsule, but this pressure is zero because proteins are not filtering from glomerulus, so it is 0 mm of Hg. These are the two pressures which are forcing the glomerular filtration, but there are two pressures which preventing the filtration. What are? Because we have a lot of proteins, the globulins, albumins, hemoglobins, right? And they are trying to prevent this filtration. For example, from here, at preventing the filtration, and this is called colloidal osmotic pressure. Colloidal osmotic pressure in glomerulus, right? So here, minus first pressure is colloidal osmotic pressure in glomerulus, and here colloidal osmotic pressure in Bowman's capsule. This is zero, but this is not zero. This is about 35 mm of Hg, right? Plus another pressure. Here, for example, the water is filtering from glomerulus, and this is funnel like. I have told you that this structure is like a funnel. So when this area is filled of water, for example, if here we have a hydronephrosis, or we have kidney stones and nephrons, the water will be retained in Bowman's capsule. So these water are responsible to prevent the glomerular filtration, and this pressure is called capsular hydrostatic pressure. Capsular hydrostatic pressure. This is glomerular hydrostatic pressure. This is in Bowman's capsule, so it is called capsular hydrostatic pressure. This is because of water, so it is called hydrostatic, and this is also because of water, so it's called capsular hydrostatic pressure. So capsular colloidal osmotic pressure plus capsular hydrostatic pressure, which prevents the filtration. This is about 10 mm of Hg, right? Now this is when we add 55 m0, it is 55 mm of Hg minus 35 plus 10, it is 45 mm of Hg. So when we subtract 45 from 45 from 55, we will get 10 mm of Hg. So this all glomerular filtration, which is called ultrafiltration, right? I have told you urine formation completes in 3 steps. First one is glomerular filtration. This glomerular filtration occurs under the pressure 10 mm of Hg. 10 mm of Hg. This is the net filtration pressure. Right, this is the first thing in urine formation. That in urine formation, the first step in which the large substances are filtering from glomerulus or occur under the pressure of 10 mm of Hg. and the osmolality of this blood is 300 milliosmol per liter. Right, this is the second thing. The third thing, the rate at which this filtration occurs is called glomerular filtration rate. Glomerular filtration rate. The first step in glomerular filtration is the net filtration pressure, which is 10 mm of Hg. The second thing is GFR, glomerular filtration rate. Specifically, it is 125 ml per minute. But in medical, I have told you that we have range. We have minimum and maximum range. So this glomerular filtration rate is from 90 to 125 minutes per liter. This filtration occurs at the rate of 90 to 125 ml per minute. This is called glomerular filtration rate, and this process is called glomerular filtration. Right? In glomerular filtration, which things leaves the glomerulus? First, water. Water will leave the glomerulus. Second, sodium or electrolytes. And electrolytes, we have a lot of sodium, we have potassium, we have HCO3 minus bicarbonate, we have a lot of calcium, magnesium, chloride ion. Right? And also we have nutrients. Nutrients are also filtering from glomerulus. Nutrients. In nutrients, we have glucose, amino acids, lipids, and small proteins like insulin and vitamins. Nutrients are also filtering from glomerulus. The next are the waste products coming from blood, like for example in the result of metabolism in body, are also drugs. Some of the drugs also coming to glomerular filtrate. So first of all, we will discuss this glomerular filtration, and I have discussed this glomerular filtration, right? But today we will only took a look on this, or we will repeat this glomerular filtration in shortly, right? So these are glomerular pressures or net filtration pressures, and these are the things which are filtering during glomerular filtration. Now we will take this is ducts. This is, for example, collecting duct, right? And this is the whole nephron. And here we have, this is, for example, the interlobular artery, and from here we have efferent arteriole, and efferent arteriole is making glomerulus, right? And this blood will be divided main and tubular capillaries, right? Here also we have blood capillaries. These are blood capillaries, and what is the purpose of these blood capillaries? The absorption and reabsorption occur in blood, right? Here also we have blood capillaries, right? What is this part? This part is called Bowman's capsule. This is called Bowman's capsule. This part is called glomerulus, right? This part is called proximal convoluted tubules. This is called loop of Henle. This is distal convolute collecting ducts. Right, this is the whole nephron. Now from here, the blood is coming to kidneys. How many blood is coming to kidney? Of cardiac output 20%, and 20% is about 1000 ml. So here 20% of blood is coming into the whole nephron. The whole nephrons mean to both kidneys 20% of the blood from cardiac output is coming. It means that this 1000 ml blood is dividing in 2.4 million nephrons, right? But here we have one nephron, so 1000 ml blood is coming here. This blood have a lot of things like electrolytes, it have water, it have cells, it have proteins, glucose, etc. Right? And these all things are passing from glomerulus. Out of this 1000 ml blood, 40% is are generally, when we say that this is plasma, and plasma, not plasma, blood, and blood we have 60% plasma, and we have 40% cells, cellular parts, right? This 40% cannot cross the glomerulus capillaries, right? So 40% of the blood will be passing from an unchanged form, mean 400 ml, right? Out of 600 ml or 60%, only 20% is filtering from, for example, this is 600 ml plasma. But plasma protein cannot cross the glomerular basement membranes and penetrated capillaries, also the Bowman's capsule walls. So only 20% of the blood is filtering from this glomerulus. Clear your concept here. 20 or 22%, which is about 125 ml. 125 ml, and that's why we are saying that glomerular filtration rate is 125 ml, because this kidney getting 20% of the blood from cardiac output per minute, and per minute it only filter 125 ml. So glomerular filtration rate is 125 ml, right? This step, the first step is glomerular filtration. This step is glomerular filtration and urine formation, right? During glomerular filtration, we have a very important basement membrane here, glomerular basement membrane. From here, the first thing which is filtering is water, right? The second, electrolytes. Which type of electrolytes? The important ones are, first, sodium, potassium, calcium, magnesium, chloride and bicarbonate ions, right? These are the electrolytes which are filtering from glomerulus, right? Next thing which is filtering from here, from blood into Bowman's capsule, is nutrients. Which type of nutrients? For example, glucose, amino acids, lipids, and some of the vitamins, right? The next thing filtering from here is drugs, toxins, and some of the waste products also, right? And important thing here, one which we are saying that proteins are not filtering from glomerulus, but small protein can filter from the small proteins like insulin. This whole thing is called glomerular filtration. This is the first step in urine formation. The first step, glomerular filtration. And why it is called ultrafiltration? Because all the things filtering from blood into Bowman's capsule. Ultra mean large, all the large things which are present in like glucose, amino acid, lipids, etc. They are filtering from glomerulus. This is the first step. Now, here in proximal convoluted tubules, we have a special types of cells. What is the name of that cells? Anyone? Cuboidal epithelium, but having brush borders on lumen side of the nephron. So for example, these are the brush border cuboidal epithelium, and here we have blood stream. This is blood, right? All these things are coming to proximal convoluted tubules. Here in these cells, now you will tell me a special type of ATPase is present in all these cells. What is the name of these ATPases? Sodium potassium ATPases. What is the role of these ATPases? It takes sodium from inside and gives potassium, right? For example, here this is one cell, proximal convoluted tubules, right? And here we have, for example, these are the sodium potassium ATPase, and these sodium potassium ATPases are occurs on all the cells of nephron, right? It takes 3 sodium from inside and gives 2 potassium 2. It means that sodium concentration in the cell is decreasing, right? And it makes a concentration gradient here, right? So what happens? First of all, here we have a very special transporter called sodium glucose antiporter. Sodium glucose antiporter. Here we have transporter called sodium amino acid cotransporter. Here we have, and these are called sodium hydrogen antiporter. So these are different types of transporters here. From here it takes the glucose plus, for example, it takes glucose, and with glucose it also move the sodium. This is called the sodium glucose cotransporter, right? Like for example, we have amino acids and sodium. It takes amino acids and sodium into the blood from proximal convoluted tubule. These are this channel is very unique, and this is called the sodium hydrogen antiporter. That means that here the secretion also take place. What is the secretion? It takes the sodium from the lumen of this nephron into the blood, but from here it takes the hydrogen ion here. So it is called antiporter. Sodium hydrogen antiporter, because sodium is getting into the blood and proton is coming from the blood. So it is called antiporter, right? Next thing is lactate. Lactate is also moving with glucose. Chloride ion. Here we have a very important reaction take place, which is carbon dioxide plus water, in the presence of carbonic anhydride. hydrase and it forms H2CO3, and HCO3 is breaking down into Raza bache this is sodium, potassium, sodium, hydrogen and tea powder, right? Here it converts into hydrogen ion and HCO3 minus, right? Now it is dependent. When the pH of the blood is low, this bicarbonate will goes into blood, and when the pH of the blood is increased and the acidity is decreased, this hydrogen ion will go to blood. But this is absorption and reabsorption processes. So the second step in proximal convoluted tubules is reabsorption, and reabsorption, we have reabsorption of water, we have reabsorption of sodium, we have reabsorption of chloride ion, we have reabsorption of magnesium and calcium. Now for example, this is potassium ion, the hydrogen ion, this is hydrogen ion. This hydrogen ion is coming into the lumen, and from here sodium is leaving the lumen. This is the antiporter system. This hydrogen ion coming to lumen and this sodium goes into the cell, and this sodium will leave in the form of this co-transporter, right? And this bicarbonate ion when came here, it gives HCO3 minus, it gives chloride ion and back into the cell. This is the reabsorption of chloride. Now what are the values of the reabsorption in proximal convoluted tubule? The second step is reabsorption. The first reabsorption occur in proximal convoluted tubules. In proximal convoluted tubules, we have the reabsorption. First of all, water. Water is reabsorbed 65%. Sodium 65%. Chloride ions 50 to 60 or 55%. Calcium 60% reabsorption take place. Magnesium is not clear yet. How many magnesium is reabsorbing from proximal convoluted tubules, but this is up to 7%. Up to 7%, right? And bicarbonate ions HCO3 minus reabsorbed, this is 90%. 90% of bicarbonate, because here also bicarbonate ions are coming. So bicarbonate ions are reabsorbing, but the reabsorption of bicarbonate ion is indirectly, because for example here I have this hydrogen ion, and this bicarbonate ion is coming here and react with this thing and will convert into H2CO3, and quickly it will again convert into CO2 and H2O, and this bicarbonate ion will leave the cell, right? I have discussed the mechanism behind in proximal convoluted tubules, right? So these are the processes of reabsorption occur in proximal convoluted tubules, and these are the values to which the things are reabsorbing. Now you will tell me the reabsorption of glucose is 100%, amino acids 100%, lactate 100%, lipids 100%. But when the lipids are reabsorbing, the urea is also reabsorbing with lipids, right? So these are the processes. If you want to show it here, first of all, water is reabsorbing sodium, magnesium, potassium. What is the process for magnesium and calcium? Magnesium and calcium transport occur paracellularly. For example, this is calcium and magnesium. They are traveling in between the cells. This is the reabsorption, right? Now we are going for loop of Henle. In loop of Henle, this is the descending limb, and this is, for example, the blood supplies. Vasa recta, like vasa recta. So from here we have which type of transporters? Aquaporine type 1. Aquaporine type 1. A lot of aquaporine type 1 are present in the descending limb of loop of Henle, right? And from here only the reabsorption of water taking place, right? So here, for example, these are water is reabsorbing from descending limb of loop of Henle, right? So this is the reabsorption of water. In which part? The reabsorption second step occur in loop of Henle. And loop of Henle, first of all, water is reabsorbed. How many water is reabsorbed here in loop of Henle? 15%. 15%, right? So here 15% of water is reabsorbing. Here 65% of water is reabsorbing in proximal convoluted tubule. Now when these filtrate are coming to this loop of Henle, it have 20% of water. And how many sodium is present here? Subtract 65 from 100, 35. 35% is remaining. So 35% of sodium is remaining, right? I want to mention it clearly because these things are important. From here, for example, this is the loop of. What happens to the osmolalities here? Osmolality will be increasing because the solute concentration is increasing, because this descending limb of loop of Henle is impermeable to electrolyte. Here we have remaining water is 20%, sorry. Remaining water is 20%. Remaining sodium is 35%. Remaining chloride ions is 50%. Remaining magnesium ion is about 80% remaining, right? And calcium, remaining calcium is about 40%, and remaining bicarbonate ion is 10%, etc. And 0% of glucose, 0% of amino acid, and 0% of lipids are coming here. It means that water concentration is very low as compared to these electrolytes. So the osmolality of this area will be high, which is about 1200 milliosmol per liter. 1200, and this solution is hypertonic as compared to blood, right? So all these things are coming to the thick part of loop of Henle, but this part is very impermeable to water, and it is very permeable to chloride ions and electrolytes. Here we have a very special transporter. What was the name? Sodium potassium 2 chloride co-transporter. And I have told you that this is very important because in pharmacology you will see that the antihypertensive drug, which is called the loop diuretics, are acting on this transporter, right? For example, this is the transporter, and this transporter is sodium, potassium, 2-chloride co-transporter, and by this which thing is coming to blood? The sodium, the potassium and 2-chlorides coming to blood, reabsorbing, right? So from here how many sodium is reabsorbing? 25%. And how many is coming to this? 35% sodium. That means that when these filtrate is coming here to distal convoluted tubules, we have only 10% of sodium is remaining, but we have 20% of water is remaining. We have 20% of water is remaining, and this potassium is again coming to this area and making this area very positive. And when the calcium and magnesium found this area very positive, it will leave, for example this is the blood supply, it will leave this lumen into the blood, right? This is hormonal independent calcium transport which is occur paracellularly. For example, this is one cell, this is another cell, this is another cell, it occur paracellular transport, calcium and magnesium, and this transport occur due to the repelling of the potassium ions. Potassium ion concentration is very high here, so it makes it very positive plus 8 millivolts, and it repel the calcium and magnesium because they are also positive, and positive repels each other. Now this is the process occur in loop of Henle. In loop of Henle we have reabsorption of water which is 15%, but this reabsorption occur in the descending limb of loop of Henle. The second thing, potassium is also reabsorbing. Third thing, chloride is also reabsorbing, and sodium is also reabsorbing, which is about 25%. Right? These are the reabsorption processes occur in loop of Henle. Now we are coming to distal convoluted tubule. In distal convoluted tubules we have two parts. One is early distal convoluted tubule and second is late distal convoluted tubules. In early distal convoluted tubules we have reabsorption of what will happen to the osmolality when a lot of chloride, potassium, sodium is leaving the lumen. It means the osmolality is decreasing because the water concentration is increasing, because water cannot leave this lumen in ascending limb. Here we have 20% of water and 10% of sodium. The osmolality here is how many? From 100 to 200 millismol per liter. It means that the solution here is hypotonic and the solution here is hypertonic and solution here is isotonic, because the osmolality is 300 and here also the osmolality is 300. Right? Here, now you will tell me what happens to the chloride ions here. We have a very special transporter here which is called sodium chloride co-transporter. And sodium chloride co-transporter, for example, we have, this is the cell of distal convoluted tubule, and of course we have sodium potassium ATPases, so sodium is leaving and sodium is deficient now in cell. From here it takes sodium and also it takes chloride ions, and this channel is called sodium chloride symporter, or it is called sodium chloride co-transporter. Right? But here one very important process occurs which is dependent upon parathyroid hormone. parathyroid hormone, the reabsorption of calcium. How many sodium is leaving this lumen? It is about 5 to 6%. It means that reabsorption of water, sodium occur in this reabsorption of, in which part? In distal convoluted tubules. Sodium reabsorption occur 5%, 5 to 6%. How many sodium is remaining now? From 4 to 5%, 4 to 5%, because 5 to 6% is reabsorbing. From here we have parathyroid gland, parathyroid receptor, and parathyroid hormone is coming from parathyroid gland. It attacks on this and will phosphorylate the calcium channels by activating phosphokinase A enzyme. Now the calcium is leaving the lumen into the blood. I have discussed this mechanism in distal convoluted tubules in detail, but by parathyroid hormone, this is hormonal dependent calcium reabsorption. Hormonal dependent calcium reabsorption in distal convoluted tubule, and this calcium reabsorption is hormonal independent. Here the calcium reabsorption is independent, right? Independent of hormone. These two processes occur in the early distal convoluted tubule. This thing which is present in the lumen of the nephron is what? It is the urine which is forming. It is the urine which is forming. The sodium and water concentration is decreasing, right? When these things are comes to late distal convoluted tubules, this is early distal convoluted tubule. Which thing is remaining? We have sodium. How many percent? Only 5%, for example. Only 5%. Water 20%. Some of the water can reabsorb from here with this sodium, but in a very little amount, right? So water is present in a lot of amount. From here, all these things also coming to these late distal convoluted tubules. And here, for example, I have the principal cell. What is the principal cell? These are the cells of distal convoluted tubules. So here I have principal cell. And what is the role of this principal cell? Anyone? It acts on aldosterone. For example, I have nucleus, and here I have kidney, and on kidney I have adrenal gland. From adrenal cortex, aldosterone is secreting, and this aldosterone came into these principal cells, and principal cell it will attach to the aldosterone receptor. This is the aldosterone receptor, and this is the aldosterone which is binds to this receptor, and it will form three types of proteins. One is sodium potassium ATPases, one is sodium transporter, and one is potassium transporter, but not one. They make a lot. It will cover all these membranes. For example, all these greens are sodium transporters, and all these reds are potassium transporter. So it will take sodium from the lumen, and it will give potassium to the lumen, right? And we say that aldosterone is releasing in response of hyponatremia. Hyponatremia means when the blood sodium level is low, In response of this hyponatremia, adrenal gland secretes aldosterone, and aldosterone acts on principal cells, and principal cells makes a lot of sodium channels, and these sodium came into the principal cell, and by these ATPases the sodium leaves the cells and goes to blood. Now the sodium level in blood is increased, right? This is the role of late distal convoluted tubules. And last we have collecting duct, but in collecting duct we have three types of cell. One is principal cell, one is intercalated A cell, and for example this is principal cell and this is intercalated B cell and this is principal cell. The principal cells in collecting ducts and in distal convoluted tubules works on same way, right? Here this is intercalated A cell. What is the role of intercalated A cell? Acidosis. For example, this is intercalated A cell and this is the blood vessel. Now, when the pH of the blood is decreased, pH of blood is decreased, that means acidity is increased, right, and acidity is decreased. It will give carbon dioxide to the intercalated A cell, right? And here this carbon dioxide will react with water, and when it react with water in the presence of carbonic anhydrase, it will form H2CO3, and this H2CO3 will break down into hydrogen ion and HCO3 minus. This bicarbonate ion will goes into the blood, HCO3, right? And this is negative charge and this is very basic, so it increase the pH of the blood and maintain the acidity or the pH of the blood. What happens to this hydrogen ion? Hydrogen ion came into this lumen, and for example from blood ammonia is coming, and this hydrogen ion plus ammonia, they react and form a less toxic substance which is called ammonium, right? This is the role of intercalated A cell. For example, this is intercalated B cell. What is the role of intercalated B cell? For example, in blood we have high pH and acidity is decreased, alkalosis, right? What happens? It gives carbon dioxide to this cell and it reacts with water and forms carbonic acid in the presence of carbonic anhydrase, and quickly this carbonic anhydrase is break down into HCO3 minus ion and H plus ion. We have proton pumps here and it push this proton into the blood, and when hydrogen ion is increasing, the acidity is also increasing and the pH is decreasing. So the pH is decreasing and it maintains the pH of the blood, intercalated B cells in the form of alkalosis, right? What happens to this bicarbonate ion? Came into urine and will react with this hydrogen ion and will form H2CO3, and quickly it will break down into CO2 plus water, and this water will leave in the form of urine, right? These are the things which occur in distal convoluted tubules. But one thing, we have principal cells here and they are dependent upon antidiuretic hormones. For example, this is the collecting duct and this is the cell of This is the principal cell of collecting duct, and these cells have a type of vesicle, and this vesicle have a special type of aquaporine transporters. Which type of aquaporines are? Aquaporine type 2 are present in principal cells of collecting ducts. Here, for example, the water concentration in blood. For example, this is the blood. The water concentration in blood is decreasing. It will signal to the posterior pituitary gland, and the posterior pituitary gland will secrete ADH, antidiuretic hormone. This ADH is, for example, antidiuretic hormone occurs here, and it will activate the receptor, and this receptor will activate the phosphokinase A enzyme, and this phosphokinase A enzyme is phosphorylate this vesicle, and when this vesicle is phosphorylated, for example, here this is the same cell, and this vesicle is phosphorylated, it came here and will attach to the lumen side of these principal cells, right? And what will happen? The water which is present in this urine will go into the blood in response of ADH, and these are the aquaporine type 2 receptor, and here we have aquaporine type 3 and type 4 receptor. This water which is coming from the lumen of the nephron will leave these cells and goes into the blood, and the water concentration of blood is increased. This is the function of ADH. In the presence of antidiuretic hormone, the amount of urine will be low, and in the absence of antidiuretic hormone, the amount of urine will be high, right? Now, urea is also present in this urine, right? But all these areas are impermeable to urea, and last distal convoluted tubules, for example, these green cells are very permeable to urea, and when urea come in contact with these cells, it leaves the collecting duct. For example, urea is leaving collecting ducts, and the remaining urea will be excreted in urine. This urea came into the loop of Henle and reattached to or retransferred to the lumen, or for example, it came into the proximal convoluted tubule. This process, and for example, this urea is again came here and reabsorbed, secrete and reabsorb. This process is called urea recycling. Urea recycling process, right? And the remaining thing in lumen is urine. Now, what is in urine? Water, urea, electrolytes, waste product. Waste products are coming from, and waste product we have oxalate ions, urea, right? Creatinine, because in some of the secretions, create also. Here, what is the secretion? These are the reabsorption processes, and secretion, waste products, drugs, creatinine, oxalate ions, they are secreting into the lumen, and when they are secreting into the lumen, it will be excreted in urine, right? So all the things remaining in collecting ducts will go to the pelvis, renal pelvis, and from renal pelvis it will leave the kidney to urinary bladder, and from urinary bladder it will leave the body, and this is the urine formation. What are the steps? The glomerular filtration, the reabsorption process, and the secretion. This thing is called Secretion. The secretion of waste product, secretion of hydrogen ion. What are the secretions? Secretions, waste products. Waste products mean the waste metabolites which are coming from metabolism of nutrients, right? Drugs, hydrogen ion, electrolytes, and water. These are the, sorry, not water is not secreting here, electrolytes. These are the things which are secreting into the lumen and will leave this nephron in the form of urine. This all process is called urine formation. Here this transporter is called when electrolytes are coming from this and water is coming from here. This is called counter current multiplier mechanism, right? And here we have, if we see the differences in osmolarities, we have osmolarity of 300, 500, 700, 900 and 1200. For maintaining this is basically, this is the renal medulla, the pyramid, and in pyramid we have a lot of electrolytes. So electrolytes, and here water is leaving and electrolytes is coming from this area. Reabsorption and absorption of water. This vasa recta is called counter current exchanger. Counter current exchanger. Why? Because it maintains the osmolality of this pyramid, right? So this whole process is called urine formation.